When we think about huge scientific breakthroughs, like life-saving drugs or decoding our own DNA, what comes to mind? Probably brilliant scientists in shiny labs. But what if I told you the real story often stars a cast of tiny, overlooked characters? We're talking fruit flies, bacteria, and even the yeast that makes our bread. Today, we're going to meet some of science's most important unsung heroes.
So let's start with a massive problem for scientists. How on earth do you study something like cancer or Alzheimer's? I mean, you can't just run risky, unknown experiments on people. That's a complete non-starter. So what's the workaround? How do you tackle this? Well, the solution is pretty brilliant, actually. You find a stand-in. Scientists use what are called model organisms. And I love this analogy. Think of them as biological stunt doubles. They're simpler species that let researchers safely explore the fundamental nuts and bolts of life that we all surprisingly share. But not just any creature can get this gig. Oh no, there is a very specific playbook, a strict set of rules an organism has to follow to join this elite team. So let's break down what it actually takes to make the cut. Okay, to get on the team, you've got to check all 6 of these boxes. First, you have to be small and easy to take care of in a lab. You also need to grow up super fast so we can study multiple generations in a short amount of time. You've got to be simple enough to work with. And this is a big one, it has to be easy for scientists to tweak your genes. Of course, your entire genetic code, your genome, has to be mapped out. And finally, there needs to be a whole community of other scientists already studying you, sharing tools and data. Yeah, it's a pretty exclusive club.
All right, first up, let's meet the OG, the original pioneer, Escherichia coli, or as we all know it, E. coli. This little bacterium is the absolute workhorse of genetics. I mean, it basically laid the foundation for the entire biotech industry.
Just look at that generation time. E. coli can double its population every 20 minutes. That is an unbelievable superpower for a researcher. You can literally grow billions of them overnight in a tiny flask. It makes doing genetic experiments incredibly fast and efficient. And its list of achievements, it's just massive. E. coli was the organism that let us figure out how to clone genes. It taught us the absolute basics of life, how DNA gets copied and how genes are switched on and off like a light switch. And maybe its most famous contribution, it became the world's first tiny factory for producing genetically engineered human insulin, completely changing the game for millions of people with diabetes. But, it's not perfect. Its biggest strength, its simplicity, is also its biggest weakness. It's super easy to work with, but because it's a simple prokaryote, it lacks all the complex internal machinery that our cells have. So to understand those more complicated processes, the team needed a new recruit. And that brings us to our next specialist, good old baker's yeast. Yep, the same stuff that makes our bread rise and our beer. But for scientists, this single-celled fungus is like looking at a simplified mirror of our own cells. And why is that? Because it's a eukaryote, just like us. It's got the complex goods inside. Now, it's not quite the speed demon that E. coli is, but it still divides every 90 minutes. And here's a huge milestone. Back in 1996, it became the very first eukaryote, an organism with complex cells like ours, to have its entire genome sequenced. That was a massive deal. And yes, its name literally means sugar fungus of beer. Perfect. The breakthroughs that came from studying yeast are just staggering. Scientists used it to crack the code of the cell cycle, the fundamental rules of how cells divide. And since cancer is just uncontrolled cell division, you can see how unbelievably important that research was. They also discovered telomeres, the little protective caps on our chromosomes tied to aging, and autophagy, the cell's internal recycling program, which has huge links to fighting diseases like Alzheimer's and Parkinson's. So, yeast is an incredible stand-in for a single human cell. It's simple, it's safe, and it shares a shocking number of our key genes. But it's still just a single cell. It doesn't have a nervous system, it doesn't have organs, it can't tell us how a whole organism works together. For that, we need another team member. So what about plants? What about the entire plant kingdom? For that, scientists turned to a tiny, humble little weed called Arabidopsis thaliana. This plant is basically the Rosetta Stone for understanding the genetics of every other plant on Earth. I mean, look at that life cycle. It goes from a seed to a flower and back to a seed in about 50 days. That is ridiculously fast for a plant. And its genome is tiny for a plant, which makes it way easier to study. You've probably walked right past this thing 1,000 times and never noticed it, but for a plant biologist, this little weed is an absolute rock star. This plant is the undisputed champion of its field, hands down. Its small genome and quick life cycle put research on fast forward. And here's a huge bonus. It can fertilize itself, which makes doing genetic experiments so much simpler. It's also become a go-to tool for studying epigenetics, this fascinating idea of how the environment can actually flip gene switches on and off without changing the DNA code itself.
Okay, so we've met the team, a bacterium, a yeast, and a weed. But let's get to the real payoff. How does any of this actually help us in the real world?
Let's start with a number, 20%. What could 20% possibly have to do with us and a single-celled fungus like yeast?
This is just mind-blowing. It's estimated that about 20% of human genes that are involved in diseases have a direct counterpart in simple baker's yeast. Just let that sink in. It means we can study the root cause of a complex human disease by looking at a much, much simpler version of it inside a yeast cell. It's an incredible scientific shortcut.
And this is where it gets really powerful. Scientists can literally take a broken, mutated human gene, say one that causes cancer, and put it inside a yeast cell. Then they can test thousands and thousands of potential drugs on that yeast to see what might fix the problem. This can just dramatically speed up the hunt for new, life-saving treatments. So when you look around, the legacy of this team is everywhere. E. coli gave us mass-produced insulin. Research on yeast paved the way for modern cancer therapies. And everything we learned from Arabidopsis is being used right now to create stronger, better crops to help feed our planet.
So next time you see some big headline about a new medical discovery or a huge advance in agriculture, just remember, that story didn't start there. It probably started years, even decades ago, in a lab with one of these tiny, incredible, unseen heroes doing the heavy lifting. From bacteria and yeast to a common weed, these simple forms of life have taught us some of the most profound secrets about our own biology. It really just makes you stop and wonder, what on earth are they going to teach us next?